Hello, my name is Willow, and today I'm going to be talking about the Avery, McLeod, and McCarthy experiment that took place in 1944. Before I begin, I briefly wanted to discuss the Griffith experiment that took place in 1928 and dealt with the topic of transformation. In this experiment, Griffith looked at virulent S strain and non-virulent R strain of Streptococcus pneumonia. He found that when he injected heat-killed virulent S strain and non-virulent R strain into a mouse, that the mouse died and S strain was recovered. This led scientists to begin to ask how this came to be and what element of the bacteria was responsible for this outcome. This is where Avery, McLeod, and McCarthy come in. They were able to use the conclusions of Griffith's experiment and other proven scientific foundations of the time to create their own experiment and determine the chemical nature of the transforming substance. They essentially picked up where Griffith left off and took it a step farther. They first made a sample of the virulent S strain of Streptococcus pneumonia and used heat to kill the bacteria and filter the sample. They then took samples of the solutions they had just made and treated it with different enzymes that destroy proteins, DNA, and RNA. Remember, they were trying to figure out what specific part of that heat-killed virulent S strain was responsible for the transformation of it into non-virulent R strain. They treated one sample with RNase, which destroys RNA, another with protease that destroys proteins, and a third with DNase that destroys DNA. Once these three samples were created, they then added each one to pure cultures of the non-virulent R strain bacteria. They found that cultures treated with protease and RNase contained the transformed S strain, and when these cultures were injected into mice, they died. However, the culture that was treated with DNase did not contain any transformed S strain, but only the non-virulent R strain, and the mice injected with this culture survived. This experiment provided the evidence needed to conclude that since DNase destroyed the transforming substance must be DNA. This experiment was pivotal, and it paved the way for many future experiments to come. Prior to this experiment, it was common belief among many bacteriologists and scientists that genes were proteins. But it was through years of research and experimentation, like the ones I just described, that proved a substance made of nucleic acids, specifically deoxyribose nucleic acid, was the definitive transforming principle. I believe the results of this experiment helped the scientific world realize the power and complexity of DNA and push scientists to learn more about this universal life-defining substance.